Okay, some things to take into account. I fully understand that maybe you have to go around here and you're like, oh my god, I'm going to appear in the video or whatever. And if you have to go to the whiteboard to explain whatever thing, and you don't want to be on the internet showing yourself, you can tell me and I will, pixelate your face or distort your voice or whatever. I don't have a problem with that. Is it okay? Is it okay for everybody if I record this, the theoretical class? Okay. Another thing, this class is extremely, I wouldn't. It's like a real. It's like a real life experiment. I mean, this is not designed to make you do all the things. You all exactly the same. So everything is going to be okay and nice, and you are going to get everything done, and at the end you are going to see the result. That's not what we are expecting for you to learn. This class is one of the I mean, you are going to probably fail. It's the thing that happens all the years. People fail as hell. Hey, hello. So why is that? Why are we repeating this class with the fail part? Because everybody seems to enjoy discovering or maybe guessing where was the failure. I mean, you're going to do a cloning, and we will discuss what does it mean right now. But there's a lot of possibilities to make mistakes. Meanwhile, you are doing genetic engineering, and it's never obvious why the things they are not working as expected. So here, you don't have like a paper saying everything that you have to do. We're going to give you that information, and you are free to write as much information as you think that is important or not. The same applies to the results that you are going to be obtaining. You are free to write everything. The tricky thing is that, the price, if you write everything, is that you are going to be able to explain more failures at the end. But I really hope that at least some groups have a proper result, more or less. So let's start. This is no, let's not start yet. Welcome to Cell Engineering and Tissues. This is Introduction to Cloning. Can you please, some of you, anyone, can you say what does it mean, cloning? If you don't feel comfortable speaking in English, you can say in whatever language you want, and I'll try to answer, including what you say. What does it mean, cloning? Come on, what does it mean to clone something? To make a copy, an exact copy. Okay, so what does it mean, cloning? The creation of exact copies. Okay. In short, that would be something like when you have to work. Okay, better we are going to go to the next one, and when we were talking about DNA, we are going to get again that concept. Okay. Those are some examples of the things that you maybe have in mind about cell engineering or tissue engineering. That it will be more tissue engineering. This is like a matrix of polymer, organic polymer that they created, that you use it to culture some cells, and so they grow in that 3D matrix, and you have something like resembles an ear, right? This is another technique, which is decellularization. It's like removing all the cells but keeping intact the matrix that surrounds all the cells. So you can use that matrix to put new cells. Is it okay? And this is an example of ex vivo gene therapy, which I think you are more expert than me in gene therapy, because I work with bacterial cells. That would be, for instance, extracting cells from a patient, and then we modify them maybe with a adenovirus is what?
Lentivirus.
Lentivirus, okay, I'm not an expert in gene therapy. So once you've selected the cells that you want, you implant them in the body, and you expect to, see some change or whatever. More things. Okay, I'm going to tell you something. For some reason, and I don't understand, but I'm like an outlier, people want to work with mammal cells. They are cool, they are from an animal, we are mammals, and people want to work with mammal cells, and they're really excited about that, and most of the groups that work with organisms there, well, most of them, work with organisms there. They work with higher organisms, like Drosophila melanogaster, C. elegans, those are mammal cells with fibroblasts, and or the, I was going to say rabbit, or the mice, okay? Mus musculus. But I'm going to tell you something. If you want to modify higher organisms, it doesn't matter if you're working with those organisms. You are going to use bacterial cells, because if you want to modify genes, you have to cut and paste a lot of DNA, and you need E. coli, which is a typical bacterial cell, to store your DNA or to replicate your DNA, to clone your DNA. Okay, that's the point. I mean, I have a plasmid or something that we will go through that. I have a piece of DNA that I want to replicate, and I can do it, for instance, in a machine, which is the thermal cycler, like PCR. You know what PCR is? Okay, so but it costs me more money than using the bacterial cell to replicate the DNA, because I can place that DNA inside the bacteria, and if some requirements are fulfilled, then the bacterial cell is going to replicate my DNA, and it's like it has less error. It has a smaller error rate than we with our best technology. So it's very convenient to store things or DNA pieces inside bacterial cells. Anyway, so the thing is that you are going to cut genes or whatever, or pieces of DNA. You want to join them together and use this organism as a tool. You are not doing research with E. coli, you are doing research with those one, but you want to use that organism as a tool. So that's the reason, because we are going to teach you how to do research, cloning or genetic engineering using E. coli. Okay? Can you go for the next one? Okay, and now here comes a little exercise. I want you all to take a piece of paper and write your name.
Martín y Pablo. Me he apuntado el nombre más representativo del grupo.
Okay, have you written your name? Okay, pay attention, please. This is a really important exercise. I want you all to draw a line in your paper. Now, the exercise is this. The line that you've written is DNA, okay? And I want you all, without talking to each other, and without consulting your phone or whatever, to draw all the required parts to convert this piece of random DNA into a gene, bacterial gene, that expresses, I don't know, a random protein, GFP, if you want. You have like 3 to 5 minutes. I don't know. Go ahead. Okay, so now a volunteer is going to come here to the whiteboard and show us all what he or she has written, but what that person doesn't know that it was already decided as soon as he or she put her ass on the chair. So please check if there is a paper in your chairs. Oh my God, we have a winner! We have a winner. How about that? Okay, what's your name? Pablo. Okay, do you care about? Okay, nice. Take that, please, everybody. Silent. Draw us your gene. Your team genes, in fact. Wow.
Oh shit.
Okay, now can you explain us all different parts of your gene? If you feel more comfortable in Spanish, just go ahead. I'll try to tell it, translate.
Bueno, pues el potenciador, bueno, esto lo teníamos muy claro, nos sonaba de que podía haber una parte del gen, que, bueno, una parte del DNA que inhibiese o...
Wait a second. Can you hear him? Yeah? And you also? Okay, so go. Keep.
Que podía activar o inhibir el gen. Luego está el promotor.
So it's something that modifies gene expression.
Yeah.
Okay. And then is the promoter. And what? Explain that.
Es bueno, la parte que reconoce que fue un gen. Yo me acuerdo bien cómo era. Luego está el código de inicio, que es donde se empieza a traducir de lo que viene siendo el gen en sí.
The start codon, where translation starts, okay?
Y luego el código final.
And what would be this?
Nuestro gen.
So that would be the gene.
Yeah.
Okay. After the, what's here in between? The start codon.
Yo primero lo pondría más. Que empieza directamente.
Okay. Okay. And stop codon. That's that. Okay. Please an applause to Pablo. Pablo. Stay there. Please go. Okay. Now, this is like a proposal of gene. I'm going to act as I am an enzyme. Okay, and I'm going to interpret this. So, this is something that modifies expression. So I'm going to ignore this because now I'm the RNA polymerase. So I'm in charge of transcription. Thank you very much. So, because you've put here a promoter, I think most of you have, or all probably, have written a promoter, I'm going to be able to find that here there's a gene. And I'm going to start like transcribing that in RNA, right? Okay, lovely. It's so sad that you forgot to put a terminator of transcription, so I'm going to keep doing this forever. Actually, not forever, because eventually you will find a terminator of a transcription terminator. Okay, you got this? Yes. Also, here maybe I mean, you have here which is called the coding sequence of GFP, and now I am a ribosome, okay? But this is a bacterial gene, and you haven't written something that allows me to recognize if there's an mRNA there, okay? So now I'm going to show you like the correct result of this push again. I didn't want you to accidentally, okay, thank you, spoil the solution. Okay, can you see it with the, okay. So, a gene is, okay, I'm going to erase your thing, and you are going to understand it. But stay here, because you're going to see something wonderful now. That was the line, this is DNA, and I wanted you to draw 4 things. A promoter, I think most of you have drawn that. A ribosome binding site, the coding sequence, and the terminator. Okay? So, everything that is beyond or downstream, how was it?
Like, what do you want to say?
Upstream?
Upstream or downstream? But
Downstream.
like, it depends, like,
Yeah,
from
downstream.
which direction. Downstream.
So, everything that is downstream, the promoter, will be transcribed into mRNA until the terminator. Okay? So, those things are included. And all the thing that is starting from the start codon and finishing at the stop codon will be translated into a polypeptide that will be eventually, that will become a protein. And, sorry, what's your name? Thank you. Sorry? Rosanna. Okay, things that you have to know. The RNA polymerase doesn't know anything about RNA and ribosome binding site and coding sequence. That enzyme only knows about promoter and terminator, and that's that. And even, I mean, that this is what allows RNA polymerase to find a gene. So the RNA polymerase is going to bind here, and it's going to start translating, and eventually when it reaches a terminator, it's going to stop transcription. Okay? And Rosanna, please. Thank you very much. And this is, I mean, in the mRNA, there is a terminator, okay? But if you are a ribosome, you don't know anything about promoters or terminators. You only care if, in bacterial cell, okay? If you are a ribosome binding site, which attracts like the 2 subunits of the ribosome, so they both bind to the beginning of the mRNA, and they start like scanning the mRNA until they find the start codon, which is AUG, okay? Adenine, uracil, guanine, and they start the translation process, adding all the different amino acids. Okay, do you understand me? Yes? No? Okay. Until eventually they find one of the stop codons, which there are 3 of them. And so, before going beyond, I want you to take a look at the class. Can you please raise your hand if you had at least one mistake in this exercise? One mistake. And the whole class is raising their hands, so thank you very much. Another applause for him. You are exactly the same as the other. So, why are we telling you this part? Because today we are going to do some cloning. Okay, we are going to start today and we will finish tomorrow, and we'll take a look at the results like next week. You don't know that. I mean, probably you've seen this, right? Last year. Okay, but for some reason you cannot remember these 4 guys: the promoter, ribosome binding site, the coding sequence, and the terminator. The distinction between CDS, which is coding sequence, or open reading frame means that open reading frame is all that nucleotides that you have between an start codon and a stop codon, which is in frame, of course. Okay? Yes, do you understand me? Do you have any question? You can feel free to ask whatever. Okay. But when this sequence in between the start codon and the stop codon, they codify for some protein, actual protein. It's not just a chance. Oh, by chance we found this open reading frame. No, they codify for some actual protein. It's when you say that this is a coding sequence. So, and now we put those genes usually when we want to add some functionality, some new functionality to a cell. We put these genes that we designed into a plasmid, okay, a plasmid. So I want you all to try to think a little bit. What is a plasmid? First of all,
circular DNA.
Circular DNA. Very good. It's one of the things that describe it. What does a plasmid need to? I mean, a plasmid is something that we are going to be dealing with in the lab. We are not necessarily going to design our genes like a piece of DNA alone. We are going to put those 5 pieces of gene inside a plasmid, inside that circular DNA. Okay. So the question is, okay, but eventually I want that piece of DNA, of circular DNA, to put it inside my bacterial cell. So the question is, what does this circular DNA, this plasmid, need to have to be maintained over the time inside the bacterial cell? Go, say things.
A sequence that permits him to enter inside the DNA?
The DNA?
Yeah.
Not necessarily. I mean, plasmids don't really have to integrate themselves into the genome, which is what it seems that you are suggesting, right? No, they can be alone. I mean, you have like the bacterial cell with all the genome, and the plasmid can be there alone. So you want it to be at least that way, alone in the cytoplasm of the cell. What does the plasmid need to have to be there over time? Come on. Ideas.
Un inicio de replicación.
Very good. What's your name?
Edu.
Okay, yeah, you need something that allows the plasmid to replicate itself. Because if you think about this, imagine that you have a cell that has like one plasmid, okay, which is the reality when you place one plasmid inside a bacterial cell. You only have one inside. Now, this bacterial cell is going to divide, okay. If you're only one plasmid there, and you don't have the ability to replicate yourself, then one of the two cells is going to have the plasmid, and the other one is not going to have the plasmid. But if the plasmid has the ability to replicate itself, then all the cytoplasm is going to be filled with plasmid. So when the two cells divide, both daughter cells are going to have plasmid. Yes? Okay, very good. What else could be important? You don't have any clue. Okay, let's say that it's a plasmid that doesn't have, because of its origin of replication, okay? There are like not really too many plasmids inside the cell. You have like less than 10, for instance. So, you think that it's possible that when a cell divides, one of the daughter cells has no plasmid at all? Would you say that that's possible? You say yes, yes. So everybody here, you don't really know. I mean, just imagine we have if you have a drop of bacterial cells, and everyone, all the bacterial cells, they have a plasmid inside. Well, around 10 plasmid inside, or between 5 and 10, and you have millions of cells there, and they are replicate. It's not unlikely that some of them they are going to lose the plasmid. Not because they lose it, just because one of the daughter cells, by chance, they don't have any plasmid at all, right? So the question here comes: what happens with that bacterial cell? that bacterial cell doesn't have to replicate the plasmid inside, because the others, they have to replicate all the plasmid that they have inside, right? So that costs an energy, but that one doesn't have to pay for replicating all the plasmid. Now it's free. So what will happen possibly with the growth of the bacterial cell? Okay, will grow faster. Okay, that is something that could happen. It would be lovely if we could get rid of that bacteria as soon as it appears, right? So what should be added to the plasmid to prevent that thing? Antibiotic resistant, exactly. There is another. Can you go, please? Okay, so we have the origin of replication, as Edu has said. I've written here selection marker because antibiotic resistance is one of the ways that by which you can select against, select for the cells that have inside the plasmid, and as soon as the cells get rid of the plasmids, if they are growing in an environment, in a culture media with antibiotic, they're gonna die, right? So, but there's another technique, which is maybe you've placed a mutation in the bacterial genome, so they cannot produce an essential substance like uracil. Okay, uracil is used to synthesize RNA. If you don't have uracil, you cannot have RNA. If you cannot produce RNA, you are gonna die. So what's the point here? That cell, without the ability of producing uracil, if you add uracil in the media, it can grow, right? But if you put them in a media without uracil, they're gonna die, unless you place here the ura gene, okay? Or a gene that produces uracil, so cells can live. And this is like an auxotrophic restoration or something like that, okay? The cells were is that way?
So like pH.
Yeah. Yeah? I don't really know. Okay.
Yeah, it's a bacteria which has like better ability to grow. It has
I'm going to write it in Spanish. Okay. So I don't know. Sorry, you were? Ellen? Okay. Auxotroph pH at the end? Okay. Thank you very much. Okay. So if you have an organism that cannot grow, for instance, without uracil in the media, that would be an auxotroph for uracil. If you can restore that auxotrophy by placing a gene that produces an enzyme that produces uracil. So this is another way of selecting. Okay? Now, why am I telling you all this? Well, the multi-cloning site is a piece of DNA with a lot of restrictions, like restriction target for restriction enzyme. Maybe you know what a restriction site. Thank you very much. So a restriction site is a place that an endonuclease can detect in the sequence. So it is going to cut in the middle the DNA. So when you design a plasmid, you want here to have a lot of different restriction sites to be able to open the plasmid there and place wherever you want. Like for instance your gene, okay? That gene has all the parts, has its promoter, ribosome inside, its coding sequence, and its terminator has a selection marker, for instance an antibiotic resistant, and has a origin of replication, and can produce a protein when inserted, when transformed inside a bacterial cell. Go ahead. Where do I find interesting? Could I find interesting databases with relevant information? For instance, here is the registry of standard biological parts, which is the one that is used at IGEN. Do you know what IGEN is, right? Yes or no? Yes, okay. So is that is that contest in which people design crazy things using bacterial cells? I mean, for instance, I don't know, modified probiotic with modified bacterial cells to prevent cancer when you drink it because that's a reaction because we've used those genes that from another species or whatever. So all the different genes promote a ribosome binding sites, coding sequence, terminators that you can find useful and you can put inside a bacterial cell. They are here. The EcoSeq is like the database for E. coli. It has all the promoters, all the genes, all the coding sequence of E. coli. And the open wetware is like a Wikipedia, but written by scientists. So here you can find protocols. You know, we are going to do some protocols today. We don't really remember the protocols. I mean, sometimes yes, because you are doing it like all the weeks. So at the end you keep remembering what is your work, but you don't have to remember what was the microliters that you had to add to cut and DNA or whatever. It's not necessary. It's on the internet. You only have to know that you have to cut that DNA. You want to bind that DNA. You want to amplify that DNA. How to do that is on the internet. Okay, and go ahead, please. And we are going to, and maybe I can sit here, and you can use another, yeah, because I'm going to be writing.